Meiotic recombination that is critical for genetic exchange during meiosis-1 is initiated by DNA's double-strand break, DSB, followed by multiple processes of DNA repair. The exact mechanisms how recombinases such as RAD51 and DMC1 localize to DSBs remain elusive. In this paper, we identified a novel protein, meiotic recombination modulator, Mrm, as a new player for meiotic recombination, which was encoded by an uncharacterized gene, 4930432K21Rik. By mass spectral analysis, we see Mrm associates with HSF2BP and BRCA2 that recruits recombinases such as RAD51 and DMC1 onto the DSB sites. Indeed, by immune staining, Mrm co-localized with MEILB2 that stands for HSF2BP. Also, Mrm co-localized with other single-strand DNA binding proteins such as MeioB on the chromatin during meiotic prophase. In contrast, Mrm showed less co-localization with the recombinases RAD51 or DMC1 on the chromatin. Probably, Mrm functions for the removal of single-strand DNA binding proteins, and then these recombinases can be recruited. Indeed, as we show in this paper, Mrm deletion severely affected DSB repairs and male fertility. We propose that Mrm modulates the localization of recombinases to meiotic DSB sites during meiotic recombination.